we are in fat state, we are taking our diet regularly at regular time interval, and whenever we are in fat state after taking food for 6 hours, yes, you will find that blood glucose is very high, postprandial blood glucose is high, that extra glucose will be converted to glycogen. Yes, I told you the complete pathway. HMP shunt, yes, you must know at this time the same pathway which we have discussed earlier, but I will again point out. I know you remember that very easily, where you took food. After taking food in fat state, blood glucose increased, that increased glucose will be diverted toward glycogen. Yes, glycogen synthesis, HMP shunt to produce NADPH. This glucose will go to pancreas, will release insulin. Insulin will activate glycogen synthesis, will activate HMP shunt, will activate glycolysis. Pyruvate will enter to the mitochondria. My pyruvate to acetyl coenzyme. This pyruvate dehydrogenase also activated by insulin. Acetyl coenzyme will enter Krebs cycle. One acetyl coenzyme produce 10 ATP. Yes, and we found that when the energy requirement of cell is fulfilled, extra acetyl coenzyme thrown outside of the mitochondria. Clear? Clear? Now in cytosol, so many acetyl coenzyme are there. And you all know the first step in Krebs cycle was acetyl coenzyme bind with oxaloacetate. Yes, acetyl coenzyme bind with oxaloacetate to form citrate. So first step of Krebs cycle is this. Clear? You all have idea about this. We found that now what happened in fat state, you took food, blood glucose increased, that glycogen storage is full, HMP shunt produced, yes, glucose to glucose 6 phosphate, HMP shunt, NADPH produced. Yes, clear? This glucose is converted to pyruvate glycolysis. Pyruvate enter the mitochondria. Pyruvate is converted to acetyl coenzyme by pyruvate dehydrogenase. This acetyl coenzyme bind with oxaloacetate to form citrate. This citrate come out of the mitochondria. Sometimes we say citrate shuttle. Sometimes we say citrate shuttle. Rarely no question. Just even if you don't remember, it's okay. But yes, if we know that oxaloacetate bind with acetyl coenzyme to form citrate, extra acetyl coenzyme are transferred from mitochondria to cytosol with the help of citrate. In cytosol, this will produce again acetyl coenzyme. So many acetyl coenzyme present in blood. In cytosol, so this reaction is occurring in cell. This is mitochondria. Outside this mitochondria, everything is cytosol. Everything is cytosol. So there are so many acetyl coenzyme in cytosol. We found that in cytosol there is an enzyme. What is the name of enzyme? The name of enzyme is acetyl coenzyme carboxylase. Acetyl coenzyme carboxylase. What is this? CO2 join. When CO2 join, yes, acetyl coenzyme carboxylase. I told you whenever CO2 is joined, that is carboxylase, and the cofactor for carboxylase, yes, you all know. Cofactor of carboxylase is biotin. Simple decarboxylation, B6. Oxidative decarboxylation of alpha keto acid, B1. Carbon dioxide join carboxylase. And the other name of this is malonyl coenzyme. Other name I write here. Another name for the same enzyme is malonyl coenzyme synthetase. Malonyl coenzyme synthetase. Clear? What is the difference between synthase and synthetase? In this ATP is used. You all know that carbon dioxide is joined, ATP is used. Yes, malonyl coenzyme synthetase. I will write here only at the lower. Synthase, synthetase. Synthase, synthetase. Synthase, no ATP used. No ATP used. Synthetase, ATP used. Energy is used. Yes, if you pronounce synthetase, synthase means even the pronunciation require a little bit more energy, so you can easily differentiate synthase and synthetase. ATP difference is there. And I told you all synthase are lies. EC number 4. EC number 4. Yes, I told you lies. All synthase are lies except glycogen synthase. Glycogen synthase transfer a glucose molecule, and glycogen synthase was. Please do reply. Glycogen synthase was. Yes, glycogen synthase was transferase. Glycogen synthase was transferase. Very good. So all synthase are lies, mostly synthase are lies, but except few, just like glycogen synthase is transferase. Synthetase is ligase. EC number 6. EC number 6. Yes, you will remember. Yes, if I suture, after suturing I ligate. When I ligate, I require little bit energy. So ligate, if you ligate, you will require energy. So ligase, ATP is used. Synthetase, ATP is used. So we will put this in box. Question has been asked, what is the difference between lyase and ligase? Lyase, no ATP used. Ligase, ATP used. Okay, clear? Good. So EC number 6. So I leave this. So this is synthetase. So ATP is used. Synthetase, ATP is used. Clear? And we found that suppose, yes, suppose this fatty acid synthesis is going on and this carbon is radioactive. What I said, this carbon is CO2, carbon dioxide, this carbon is radioactive. Suppose this carbon is radioactive. Suppose this carbon is radioactive. Okay, radioactive carbon. So acetyl coenzyme is having 2 carbon. Malonyl coenzyme is having 3 carbon. So a 2 carbon compound is coming to 3 carbon compound. That's why acetyl coenzyme carboxylase. Yes, if you remember malonate, I told you. Yes, I will write here only. Malonate. Malonyl coenzyme are same. Malonate is inhibitor of complex second of electron transport chain. Succinate dehydrogenase. Yes, I have discussed it earlier. Also inhibitor of CPT1. What is CPT1? I will discuss shortly. For the time being, carnitine, palmitoyl transferase 1. Carnitine, palmitoyl transferase 1, CPT1. So malonate is inhibitor of these 3. Okay, so for the time being, malonyl coenzyme or malonate is having 3 carbon, and this malonyl coenzyme is inhibitor of complex 2 succinate dehydrogenase and CPT1. Okay, what is CPT1? We will discuss shortly. For the time being, yes. Now if I ask you that this malonyl coenzyme 3 carbon atoms are there, this carbon was radioactive C14, normally C12 होता है, this is C14 radioactive carbon. Now out of these 3 carbon, one carbon is radioactive and two carbons are normal. Radioactive and two carbons are normal, which were present in acetyl. So you can say that if fatty acid synthesis is going on, if fatty acid synthesis is going on in an environment where carbon dioxide is radio labeled, then out of malonyl coenzyme A, 3 carbon, out of 3 carbon of malonyl coenzyme A, 1 carbon would be radioactive. Yes or no? Yes. So now what nature did, or what is happening in our body when so much of glucose is there, yes, so much of glucose is there, we convert extra glucose into fat. I told you glucose can be converted to fat, but fat cannot be converted to glucose. Fat cannot be converted to glucose because this is irreversible reaction. Fat cannot be converted to glucose. I told you acetyl coenzyme is never substrate for gluconeogenesis. This acetyl coenzyme, yes, reach the cytosol. Now I can say, suppose there are so many acetyl coenzyme, 10, 20, 50. Out of these 50, 100 acetyl coenzyme, few acetyl coenzyme are converted to malonyl coenzyme. Now you can say that in fad state, we are having so many acetyl coenzyme and we are having so many malonyl coenzyme. Okay, clear? Now for the time being, yes, I say, even if you don't remember this radioactive, it's okay. Why I took it radioactive? Because there was a question. So I thought that it will help you to remember the concept. Yes, if you remember at this time, for the time being, if I remove this radioactive word, even then you are comfortable. Yes, if radioactive is not there, if this part is not there, yes, so I am not concerned about this. In a simple way, I say that normally what happens in our body, whenever we take extra glucose is converted to fat. How this is converted to fat? Then all the extra glucose is converted to acetyl coenzyme. Acetyl coenzyme come out of the cell, mitochondria, and in cytosol few molecules of acetyl coenzyme converted to malonyl coenzyme. In this reaction, carbon dioxide is joined. That's why the name of enzyme is acetyl coenzyme carboxylase, because in this malonyl coenzyme is synthesized. That's why malonyl coenzyme synthetase, and in this ATP is used. So you can say now in cytosol both acetyl and malonyl coenzyme are there. Acetyl is having 2 carbon, malonyl coenzyme is having 3 carbon. Suppose I ask you to synthesize butyric acid. Yes, I want to make butyric acid, or I ask you to make butyric acid. You will say, sir, I know what is butyric acid. You told me that 4 carbon fatty acid is butyric acid and the common name of all fatty acid is acyl. What I said, common name of all fatty acid is acyl. I want to make this butyric acid. You can say butyric acid as acyl coenzyme having how many carbon? 4. I have told you all fatty acid common name is acyl. Butyryl is acyl. Palmitoyl is acyl. Stearyl is acyl. So this is acyl having 4 carbon. Now I want to this. I also know the structure. You also know the structure of butyric acid. You all know CH3, CH2, CH2, COOH. I want butyric acid to be formed. So yes, in real life, yes, things are little bit complicated, but if we think hypothetically and we say I want a 4 carbon fatty acid, what can I do? I take acetyl coenzyme, second acetyl coenzyme, join 4 carbon fatty acid. Very simple. But life is not so simple. Life is not So simple, means you can't join these two acetyl coenzyme to make fatty acid. First of all, yes, we converted acetyl coenzyme to malonyl coenzyme. First we converted acetyl coenzyme to malonyl coenzyme. Now further synthesis will take place on a fatty acid synthase complex. This fatty acid synthase complex is present in cytosol. Don't get confused with this peripheral boundary. This reaction is occurring in cytosol. This enzyme is present in cytosol, and this is fatty acid synthase complex. This name of this enzyme is fatty acid synthase complex. This is multi enzyme complex. First thing, this is multi enzyme complex. Multi enzyme complex. How many enzymes are there? 6 enzymes are there. Previously we used to consider 7 enzymes, but now we say 6 enzymes are there. Yes, in this multi-enzyme complex it is a dimer. It is a dimer. Each monomeric unit, yes, is a dimer. Each monomeric unit is having 6 enzymes. Okay, and this dimer is arranged in head to tail fashion. Yes, I have made deliberately like this. One is head, one is tail. Yes, you can say, suppose this is a dimer, this is head and this is tail. Okay, so I can say two units are arranged in head to tail fashion. Head of one, tail of another. Head of one, tail of another. These two are arranged in head to tail fashion. And if I define head and tail like this, if I define that tail is cysteine end, okay, cysteine, this is pan end. Pan means pantothenic acid, cis means cysteine. Okay, don't get confused, nobody is going to ask, but you can remember this is cysteine end. Cysteine end, this is pantothenic pan end. And you will find that another subunit, this is cysteine end and this is pan end. So in these two subunit, cysteine end of one is against pan end of another. Cysteine end of one is against pan end of another, and both are having sulfur. That's why they form disulfide bond. You all know, wherever sulfur is there, it has tendency to attach with another sulfur to make disulfide bond. Okay, so you can see that these two dimers, yes, two monomeric unit are arranged in head to tail fashion. Cysteine end is attached with pan end by disulfide bond. Pan end is attached with cysteine end by disulfide bond. Okay, clear? Now, yes, acetyl, cysteine. Now, what happened? You can remember this structure in this form. This is first unit having cysteine end and pan end. This is first unit having cysteine end and pan end. Yes, this is second unit having cysteine end and pan end. And you can remember like this. Look at me. This is cysteine end, this is pan end. So this is pan end, cysteine end. So cysteine end is against pan end. Here pan end is against cysteine end. So two opposite direction. Yes, pan end, cysteine end. Pan end, lower one is pan end, this is cysteine end. In this direction, the upper one is pen end and the lower one is cysteine end. Okay, so we found that we wanted to make butyric acid, but yes, when initially came in my mind, I thought I will join two acetyl coenzyme. Acetyl coenzyme is having two carbon, so two molecules, four carbon fatty acid. But I found that life was not so simple. What nature did, how fatty acid butyric acid is synthesized, we found that acetyl coenzyme is first converted to malonyl coenzyme. So now there are both acetyl and malonyl coenzyme in cytosol. Now we found that one acetyl coenzyme comes and bind to cysteine end. One malonyl coenzyme comes and bind to pen end. Question is asked, yes, acetyl bind to cysteine. So you can easily remember C. Acetyl is having C. So question is asked, acetyl bind with cysteine, while malonyl bind with pen. So N M. Okay, LMN LMNOP. So you will remember, question is asked, yes, malonyl bind to which end, cysteine end or pen end? Malonyl bind to pen end, acetyl bind to cysteine end. So we found that we wanted to make butyric acid. In making of butyric acid, first of all, we converted few molecule of acetyl coenzyme to malonyl coenzyme. Now we were having so many acetyl, so many malonyl coenzyme, one acetyl and one acetyl and one malonyl bind to cysteine and pen end. Now what happens? Now what happened that, yes, what happened that this is cysteine end and this is pen end. Pen. As cysteine end is attached with acetyl 2 carbon. Pen end is attached with malonyl 3 carbon. Okay. Now acetyl will come to malonyl. Condensation reaction occurs. And one CO2 is, this is CO2, yes, can you see, this is CO2 is removed. Okay, so acetyl 2 carbon, acetyl how many carbon? 2 carbon, 2 carbon comes to malonyl, 2 plus 3 5, 1 CO2 removed by condensation reaction. Right here, condensation reaction. Yes, condensation reaction occur and CO2 is removed. This, yes, and compound is formed. The compound is called ketoacyl. Yes, ketoacyl. So ketoacyl group. Yes, CO2 is removed, condensation reaction, CO2 is removed and there is formation of ketoacyl. In this ketoacyl, how many carbon atoms are there? 4 carbon atoms are there in ketoacyl. How many carbon atoms are there? 4 carbon atoms are there. Yes. Now, this ketoacyl will be converted to, yes, ketoacyl will be converted to butyric acid. What is ketoacyl? Whatever I am writing, even if you don't remember, it's okay, but you have an idea, you must have an idea how this reaction is occurring. Before going to that, again I repeat what happened, because I am going to integrate each and everything. Okay, after that, yes, we took food, extra food, glucose produce HMP shunt and NADPH, extra glucose, pyruvate, acetyl come out, acetyl convert to malonyl. So now in cytosol you both have acetyl and malonyl. And this carbon, if you take this carbon radioactive, Then out of these 3 carbon, 1 carbon will be radioactive. Now we found that one acetyl bind with cysteine, one malonyl bind with pan. When condensation reaction occur, acetyl bind with malonyl and CO2 is removed. CO2 is removed. This removal of CO2 is, yes, condensation reaction. And this carbon is radioactive. This carbon is radioactive. So whatever carbon was joined here, whatever carbon was joined here is removed in condensation reaction. So I will write like this carbon, yes, yes. So means in this ketoacyl, how many radioactive carbon? If this carbon dioxide is radioactive, so in this ketoacyl, how many radioactive carbon would be there? Yes, in this ketoacyl, how many radioactive carbon? Now, what is the structure of ketoacyl? Very good. What is the structure of ketoacyl? I will draw the structure of ketoacyl. Even if you don't remember, it's okay. Again, I say, but if you have idea, then it would be really helpful. CH3, CO, CH2, COO. This is ketoacyl. Very good. Yes. CH3, CH3, CO, keto. This is keto, ketoacyl. This is ketoacyl. Okay. Ketoacyl having how many carbon? 4 carbon. 1, 2, 3, 4. So till now butyric acid is not formed. Yes, we want butyric acid. Yes, this is not butyric acid. This is not butyric acid. This is not butyric acid. This is ketoacyl. We want to convert this ketoacyl to butyric acid. How we will convert this ketoacyl to butyric acid? By 3 steps: reductase, hydratase, reductase. So first step is reductase, and I told you wherever you want reduction, you will require NADPH. And in fed state, I have already told you, in fed state so many glucose has entered HMP shunt, so many NADPH are there. If you remember HMP shunt, I told you NADPH is produced, NADPH is used for fatty acid synthesis, NADPH is used for cholesterol synthesis, NADPH is used for steroid hormone synthesis. NADPH is used to convert, yes, yes, oxidized glutathione. So wherever reduction is required, you require NADPH. NADPH is required in fatty acid synthesis. Yes, so NADPH is required in fatty acid synthesis. So in this, and these NADPH, these NADPH is used by reductase. What reductase do? Look at the screen. Yes, reductase will join two hydrogen. It will join two hydrogen and it will form, yes, it will join two hydrogen. It will form, yes, what would happen? CH3, CH3C, hydrogen, OH, H, C, H, H, COO minus. Means I joined two hydrogen. I cut this bond, join hydrogen, join hydrogen. Clear? So I found this. Clear? That is, yes, reductase, NADPH is used. NADPH is used. I am again saying that if you don't remember these steps, it's okay, but you must know reductase, first step after ketoacyl reductase. Then hydratase. Hydratase, H2 is removed. H2 is removed in this CH3, C double bond. When H2 is removed, there would be formation of double bond, yes, COO minus. And this is H, this is H. Do you agree? Yes, double bond. So this is formed. Okay, this is hydratase. This is hydratase. Okay, water is removed. Okay, now again reductase. Again reductase means joining of hydrogen, and hydrogen will come from NADPH. NADPH. And this reductase, what will it do? It will do CH3, C reductase 2 hydrogen, CH2, CH2, COOH, and this is COO minus, yes, COO minus butyrate. So you can say, you can convert ketoacyl to butyric acid by reductase, reductase. Means you join two hydrogen, yes, you join two hydrogen, okay, and yes, then after joining hydrogen, yes, HOH, yes, is formed. Hydratase, H2 is removed, then reductase, NADPH used. So NADPH is used in fatty acid synthesis by 2 reductase, and butyric acid is formed. So this is fatty acid synthesis, butyric acid formed. And if I ask you, yes, how many radioactive carbon in butyric acid? 0. You have given answer. Now this question, a low level carbon dioxide labeled with C14 is accidentally released into atmosphere surrounding industrial worker as they resume work following the lunch hour. So why they have given following the lunch hour? You all know, after lunch blood glucose will increase, extra glucose will be converted into fat. Means after lunch hour fatty acid synthesis will occur in these persons, and C14 is accidentally released in the atmosphere. Unknowingly they breathe the contaminated air for 1 hour. 1 hour they breath, means CO2, radioactive CO2 is also there. Fatty acid is being synthesized. When fatty acid is being synthesized, malonyl coenzyme is being synthesized. This malonyl coenzyme will have radioactive carbon. If 3 carbon are there, 1/3 carbon are radioactive. Yes, 1/3 carbon are radioactive. Which of the following compound will be radioactive? All carbon atom of newly synthesized fatty acid, no carbon of, yes, newly synthesized fatty acid would be radioactive. No carbon in fatty acid synthesis, yes, will be, fatty acid will be radioactive. Half of carbon only synthesized, no. In fatty acid, no carbon is radioactive. About 1/3 carbon of newly synthesized malonyl coenzyme would be radioactive. 1/4 carbon of newly synthesized malonyl coenzyme, no, 1/3. Yes, yes, industrial worker were doing their work after lunch hour. Yes, after lunch hour, after lunch, when we take lunch, we are in fat state. I told you extra carbohydrate are converted to fat, and during that, after lunch, accidentally carbon dioxide released, which was having radioactive carbon, so when fatty acid is being synthesized and this radioactive carbon is there, so malonyl coenzyme, out of 3 carbon, these acetyl normal carbon, but malonyl coenzyme radioactive carbon, yes, out of 3, 1 would be radioactive. So they ask that after lunch hour, if accidentally CO2 is released, patient will breathe that. And yes, it's not the duration, 1 hour, 2 hour, they try to confuse you. Okay, yes, they breathe the contaminated air, that is more than sufficient. Okay, means this radioactive carbon is coming in the form of CO2. Okay, oh, so one third of the carbon of newly synthesized malonyl coenzyme would be radioactive. Clear? Got the point? So this is fatty acid synthesis. In first step, how many carbon atoms are joined? 2 plus 2, 4 carbon atoms are joined. But you said, sir, no, I want 6 carbon fatty acid. I said, okay, don't worry, we can make 6 carbon fatty acid. What I will do, I will draw it again. This is unit 1, cysteine, SH, unit 2, pan, SH. Now I will transfer this butyric acid to, yes, I will transfer this butyric acid to cysteine. I transfer this butyric acid to cysteine, means butyric acid. Butyric acid. Now this pan SH, I attach pan SH with malonyl. Malonyl. Okay, so butyric acid, cysteine, malonyl, pan. Butyric acid, cysteine, attach malonyl, pan. So now, yes, this was transferred to cysteine. This was transferred to cysteine, means cysteine is having butyric acid, pan is having malonyl. Butyric acid is having how many carbon? 4. 4 carbon. Malonyl coenzyme is having 3 carbon. Now again condensation reaction occur. 1 CO2 is removed. 1 CO2 is removed. So this butyric acid again come to this malonyl coenzyme, and there would be formation 1, 2, cysteine SH, pan SH. This, yes, CO2 will be removed, and this pan SH would be containing keto acyl. Keto acyl having how many carbon? 6 carbon. Yes, this will come to this keto acyl having how many carbon? 6 carbon, because 1 CO2 is removed. 1 CO2 is removed by condensation reaction. Clear? And this keto acyl having 6 carbon will be reductase, hydratase, reductase, and 6 carbon fatty acid. So we say fatty acid synthesis in each cycle. Yes, butyric acid synthesis is common step for all fatty acid. We say yes. Okay, let me explain again. Till now you know you made butyric acid. Till now you know, yes, till now you know butyric acid is formed. Till now you know butyric acid is formed. Yes, now what happened that this butyric acid, yes, till now you know butyric acid is formed. Now you want to make 6 carbon fatty acid. 6 carbon fatty acid. So I transferred this butyric acid having 4 carbon to this cysteine end. When I, yes, transfer to cysteine end. So now you can write that cysteine is having butyric acid. Yes, means after synthesis of butyric acid, further reaction will take place. What is the further reaction? This acetyl is transferred, oh sorry, acyl having how many carbon? 4. So butyric acid is transferred to cysteine end. Butyric acid is transferred to cysteine, and in cytosol there are so many malonyl coenzyme. One malonyl coenzyme was used here. Second malonyl coenzyme will come and will bind to which end? Pan end. So all these reactions are occurring in cytosol. Butyric acid was synthesized in cytosol. So now cysteine end is having butyric acid. Pen end is free. Malonyl came and bind to pen end. Now pen end is having malonyl. Cysteine is having butyric acid. Butyric acid is having 4 carbon. Malonyl coenzyme is having 3 carbon. 4 plus 3, 7 carbon. Condensation reaction occur, CO2 is removed. Condensation, same thing. Means if I say, okay, forget everything. If I say this is butyric acid, then what will happen? If this is butyric acid, condensation reaction occur, malonyl, butyric, 4 carbon, 3 carbon, yes, 4 carbon, 3 carbon, 7 carbon fatty acid, 7 carbon compound, ketoacyl synthase, CO2 is removed, ketoacyl compound is formed, in this ketoacyl, how many carbon should be there? 6 carbon should be there. Then reductase, 6 carbon, reductase, hydratase, reductase, and then acyl coenzyme fatty acid having how many carbon? 6 carbon is produced. So in this way, ketoacyl having 6 carbon is formed. Yes, ketoacyl having 6 carbon is formed. Then reductase, hydratase, reductase, and fatty acid is formed. Yes, every time you are joining 2 carbon, 2 carbon, 2 carbon, 2 carbon, 2 carbon. So yes, we can make even chain fatty acid. When we want to make odd chain fatty acid, we can do alpha oxidation. Alpha oxidation I will discuss. In alpha oxidation, you can remove 1 carbon. Alpha oxidation, we can remove 1 carbon. Okay, yes, alpha oxidation we can remove 1 carbon. Yes, so if you want to make odd chain fatty acid, mostly in our body they are even chain fatty acid, but if you want odd chain fatty acid, you can remove by alpha oxidation, you can produce. Okay, so in this way, how questions are asked. Even if you don't remember so much, what are the question you must know? First question is condensation reaction. Condensation reaction require, so the enzyme is ketoacyl synthase and CO2 is removed. The same CO2 is removed which was joined here. Take one. Second question, acetyl bind with cysteine, malonyl bind with pan. Okay, then sequence of enzyme reductase, hydratase, reductase. This is more important. Okay, and in this way, in each cycle 2 carbon atoms are joined. Yes, I can write, yes, caproic acid. After, yes, means in each cycle of fatty acid synthesis, 2 carbon atoms are joined, except first cycle where 4 carbon butyric acid is formed. Okay, yes, so in each cycle fatty acid synthesis 2 carbon atoms are joined, except that first cycle where 4 carbon butyric acid is formed. Okay, got the point? Yes, although in first cycle also 2 carbon atoms are joined to 2 carbon atom, but yes, this question is asked. This was a question of PGI Chandigarh actually, that in first cycle how many carbon atoms are joined. Actually in first cycle also 2 carbon joined to 2 carbon, but the resulting compound is 4 carbon fatty acid. So we started saying that the yes, first cycle 4 carbon atoms are joined. Yes, we started saying 4 carbon atoms are joined in first cycle, and further every cycle 2, and most common fatty acid synthesized by this way in our body is palmitic acid. Most common fatty acid is palmitic acid. Question is asked, which is most common fatty acid synthesized? No dear, this is ketoacyl is not fatty acid. Ketoacyl would be converted to reductase, hydratase, reductase to caproic acid. Clear? Where 4 carbon butyric acid is formed in first cycle, butyric acid is formed. How many carbon? 4 carbon. So actually we say that in first cycle, whatever I have discussed here, in first cycle you find fatty acid, how many carbon? 4 carbon. How many carbon? 4 carbon. So although 2 carbon joined with this 3 carbon, and you can say acetyl joined with 2 carbon, so in first cycle also 2 carbon joined with 2 carbon. So we should say in first cycle 2 carbon atoms are joined, means each cycle 2 carbon atoms joined. But we said no, in first cycle 2 carbon joined with 2 carbon, so total 4 carbon joined. It was really not it's not what is the not was not a good explanation, but few standard textbook has written this in this way, that in first cycle 4 carbon butyric acid is formed, in other cycle each cycle 2 carbon atoms are joined. So in first cycle how many atoms are joined? Total 4, because 4 carbon fatty acid is formed. Most common fatty acid synthesized in our body is palmitic acid. How many carbon atoms are there? 16. Clear?